Gregor Mendel, the father of modern genetics, not only researched pea plants, but also carried out several experiments with honeybees. In fact, by 1865, Mendel had published his work on inheritance of characters. However, his work did not receive due recognition until the beginning of the 20th century due to several reasons. Firstly, Mendel's work couldn't be widely publicized because means of communication in those days were not well developed as they are today. Secondly, Mendel had considered genes or factors as discrete and stable units responsible for controlling the expression of traits. He had also explained the concept of alleles and how they were responsible for the continuous variations observed in nature. However, his concepts about genes were not readily accepted by his colleagues. Thirdly, Mendel, for the first time, had applied statistical analysis and mathematical logic to problems in biology, which was objected to by many biologists of his time. Finally, Mendel could neither explain what genes were made of, nor could he provide any physical proof of their existence. Mendel's work got due credence only in 1900, when his results on the inheritance of characters were independently rediscovered by three scientists: Hugo de Vries, Carl Correns, and Erich von Schermach. Also, both chromosomes and genes segregate during the process of gamete formation, and only one of each pair is passed on to a gamete. However, they differ in the way they segregate. In genes, independent pairs segregate independently of each other, while in chromosomes, one pair segregates independently of another pair. Let's now study the behavior of chromosomes during cell division. In the anaphase stage of meiosis I, a set of chromosomes aligns along the metaphase plate. Here, you can see a long orange and a short green chromosome, as well as a long yellow and a short red chromosome aligned at the same pole. Now, consider another set of chromosomes aligned along the metaphase plate during the anaphase stage of meiosis I. Here, there is a long orange and a short red chromosome and a long yellow and a short green chromosome arranged at the same pole. Now, during the anaphase stage of meiosis II, you can see the orange and green chromosomes segregating together in the cell on the left, while the orange and red chromosomes segregate together in the cell on the right. Sutton analyzed this segregation of chromosomes during meiosis and concluded that chromosomes have individuality and they occur in pairs, with members of each pair contributed by each parent. He also concluded that the paired chromosomes separate from each other during meiosis, and the distribution of the paternal and maternal chromosomes in each homologous pair is independent of each other. Both Sutton and Boveri were of the opinion that the pairing and separation of a pair of chromosomes would result in the segregation of a pair of genes or factors carried by them. Their hypothesis on chromosomal behavior came to be known as the Boveri-Sutton chromosome theory. In fact, Sutton combined this hypothesis of chromosomal segregation and Mendelian principles and called it the chromosomal theory of inheritance.